So now we're going to talk about genomic imprinting for a few mammalian traits. Only a few now. The phenotype depends on which parent passed along the alleles for the traits. This variation in phenotype is called genomic imprinting, and it actually involves silencing of certain genes depending on which parent gave those genes. So please take a look at figure 15.17. It's usually a result of methylation. Now remember, a methyl group is CH3, so if you add these to your structure, then we call that a methylation, and it's of cysteine nucleotides. It may affect only a small fraction of mammalian genes, but interestingly enough, most of these are critical for embryonic development. Extranuclear genes, or they're also known as cytoplasmic genes, are found in the organelles in the cytoplasm. Some organelles that can carry these genes include mitochondria, chloroplast. Other plant plastids can carry small circular DNA molecules. And interestingly enough, since the zygote cytoplasm always comes from the egg, all of these extranuclear genes will be inherited from the mom, inherited maternally. One important one is a defect in mitochondrial genes that keep cells from making enough ATP. And this results in diseases that affect muscular and the nervous system. An example of that might be mitochondrial myopathy and Leber's hereditary optic neuropathy. Now, this is also with chapter 15, and we want to talk just a little bit more about the chromosomal basis of sex, because even though humans and mammals have two types of sex chromosome, X and Y, and by the way, this is known as the X and Y system. XX is female, XY is male. Remember, we already said the genes that are located on either sex chromosome are called sex-linked genes. The genes on the Y chromosome are Y-linked. Genes on the X chromosome are X-linked, and we talked about some examples of those. I have drawn out for you the way that these might be inherited. There's also a YouTube video that I posted on how to work one of these problems, okay? So you'll notice, if a normal man, X, big HY, marries a woman who is a carrier, let's say for hemophilia, and she is X, big H, X, little h. Notice their gametes, and then you also can see the Punnett square, and so what you're going to end up with is 25% completely normal female, 25% completely normal females who are carriers, 25% normal males, and 25%, well, depending on what the condition is, I should have said hemophiliac, but if it's color blindness, it would also be 25% color blindness. The female needs 2 copies of the allele in order to fully express a recessive X-link, and we call that homozygous. That means she's got to be the same in the zygote. The male only needs 1 copy of the allele, remember, to show the recessive X-linked trait, and we call that hemizygous. We already talked about the bar bodies. You can look at the example in figure 15.8. There's another system called the XO system. And examples of organisms that have this system include things like grasshoppers, crickets, cockroaches, some other insects. And interestingly enough, they only have one sex chromosome, which is an X. If you are a male, you have one X. If you're a female, you have two X's. The male produces gametes with X or without, which is a 0, a sex chromosome. The sperm then determines the sex. There is also a ZW system found in some birds, some fish and crustaceans, some insects including butterflies and moths, and some reptiles including Komodo dragons. It's kind of interesting, boas and pythons as a family group use the XY system just like we have. Females are ZW, males are ZZ. So this is kind of interesting because this time it is the egg, the ovum, that determines the sex. The Z chromosome is larger and has more genes. Then we also have what is called the haplodiploid system. And this is common in bees and ants and wasps. Males develop from unfertilized eggs, and these are haploid. So all the sperm are identical. Females develop from fertilized eggs, and they are diploid.